Duchenne muscular dystrophy is the most common of the muscular dystrophies and is caused by a defect in the dystrophin gene. But defects in more than 100 other genes can lead to different forms of muscular dystrophy, some just disabling, others fatal.
One of the genes that I know is prominently involved is called dystrophin. What does it do?
Well, the dystrophin gene causes the most common form of muscular dystrophy, known as Duchenne muscular dystrophy. It's a gene that makes a protein that's very important to hold muscle cells together. So when that gene is not working properly, you don't produce a protein and muscle cells become very fragile. And as a result, they're breaking down. They can repair themselves throughout life, but you get this vicious cycle of muscle breakdown, breakdown, and repair. And unfortunately, the repair process starts losing out, and the patients gradually lose muscles throughout their life. This slide shows an overview of a variety of different proteins that are important in protecting muscle cells from developing muscular dystrophy. Proteins that can be located outside the cell, in the cell plasma membrane, or inside the cell can all lead to different types of muscular dystrophy when those proteins are not produced properly. On the outside of the cell, a particular type of collagen known as collagen 6, when defective, can lead to Bethlem myopathy. Another extracellular matrix protein known as laminin is defective in different types of congenital muscular dystrophies that affect children at a very early age. The dystrophin protein shown here on the inside of the muscle membrane is defective in Duchenne and Becker muscular dystrophy, and this represents the most common form of muscular dystrophy. The dystrophin protein plays a key role in linking several intracellular proteins to the extracellular matrix, and a lot of those other proteins involved in that linkage can be defective in other types of muscular dystrophy. In particular, the sarcoglycan proteins are defective in a variety of different types of limb-girdle muscular dystrophy. And there are also a variety of glycosylases that are not shown here that are defective in many other forms of limb-girdle muscular dystrophy. The point here is that there are many different types of muscular dystrophy, and the proteins that are involved in these different types of muscular dystrophy often work together in common pathways that are important for protecting muscle cells from breakdown.
Many other human diseases are caused by defects in different genes. And with the knowledge obtained from the Human Genome Project, we are learning more about which genes they are and what they do.